So next we're going to hear from Tom Langbehn. Tom is a quantitative marine ecologist with a strong interest in evolution and global change ecology. Enjoys thinking broadly, linking data with theory and models, with interest in diverse biological disciplines. The central aspects of Tom's work are forging interactions among physiology, life history theory, species distributions, and behavioral ecology. Particularly fascinated by polar ecosystems and their extreme seasonality, as well as life in the ocean twilight zone. And he works mostly, but not exclusively, on mesopelagic fish, zooplankton, and birds. Tom's research combines numerical models rooted in evolutionary and ecological theory with observations from the field. And although most of the work is done, he says, from the comforts of his office, he does regularly participate in research cruises to collect data in order to ground truth and model predictions. Tom completed his PhD at the University of Bergen in 2019, and currently a lecturer there in the Department of Biological Sciences. Tom.
Hey. Yeah, thank you so much for this kind introduction. I'm very happy to be standing here. If Amanda is a social science girl in a natural science world, then I'm one of this rare breed of modelists that goes out to sea and collects data. And I hope I can invite you to join me from studying scientists to studying fish again. I will be talking about how we can use models, and specifically mechanistic models, to understand cause and consequences of diel vertical migrations. And I like to acknowledge that these are, of course, not only ideas that manifested in my head. So there are lots of other people that have been involved in this, and Stein Cartwright, I think, is the only one who's in the room today. Not sure if some of the others are joining online, but yeah, these ideas are really a group effort. And I want to start by borrowing Tracy's words. I'm going to start with the obviously blinding that diel vertical migration is really ubiquitous. So we see diel vertical migrations in all ocean basins, but what we often forget is that it is highly variable. So if we look, then we see there are areas where diel vertical migration is relatively shallow, while in other areas we see deep diel vertical migration. There are often several 100-meter differences, the daytime depths that these organisms migrate to. And there's also a huge variability in the proportion of migrants. And I think one of the key take-home messages already from this slide is that we often tend to study this phenomena on the basis of aggregation. So we look at scattering layers, but we often forget to look who are the organisms that migrate. And even more rarely, we look at individual levels, which also means we don't know if the 90% migrating is 90% of the species, 90% of the individuals, 90% of one certain size or year class, for example. So there are a lot of things that we don't understand when it comes to diel vertical migration. One approach to tackle this, and I think we have heard this already several times during the last days, is the call for we need to collect more observations. But we have a whole lot of observations, and if we start collecting observation, then this leads us often down the path that we fit statistical models to this data to try to make sense of what we have observed. I'm going to suggest a complementary approach, and this might seem counterintuitive to some of you, but we can actually do modeling without any data. You're laughing. Good. So these are often mechanistic or process-based models. So they are informed by patterns, and they're built on theory. And if I talk about theory, this is more the solidified ecological knowledge that we have. And we can bake this into equations, and we can use this as the basis to build models. There are some really benefits to this approach, and one is that it's very good to generate hypotheses that later help us when we go out and sample. We can make testable predictions, so we are not starting from scratch every time we go out and sample. And I think the real plus here is also that the units in these models, they have actually a real physical and measurable properties, so other than statistical models. So what we typically do with these models is that we don't fit them to data, but we confront the model predictions with data. And as I already said, this can already at this stage help us to inform our sampling approach. When we confront the models with the observations, we often end up in a scenario, or most often we end up in a scenario where we see that it doesn't match reality at the start. But I think this is a plus, because this is actually the opportunity to learn something. If we build models that predict reality from the start, then we have done something wrong because we already understood the system. I think it's often at that point when the models predict something different than what we would have anticipated. That's where we can refine our model parameters, we can learn something about the system. And then we can enter a cycle where we do modeling, observation modeling, and we start building understanding. I'm going to show you one example that I'm very familiar with because I've coded this model. So this is a simple model. We will walk through a few steps here, but that predicts the diel vertical migration behavior of mesopelagic fish. In the model, we have three agents. We have one piscivore, a larger predatory fish. We have a planktivore, which is parameterized to resemble Benthesema glaciale, the glacial lanternfish, which is one of the dominant species in the North Atlantic, and zooplankton at the lowest trophic level. And the interactions between those three species groups, they're realized in the model as visual encounter rates. So we know how often a predator will encounter a myxophyt or a myxophyt will encounter a plankton, and that depends on the light field, but also on the water optical properties, so how quickly light is attenuated through the water column. For the prey encounter with zooplankton, there's an additional density-dependent term, so there's a handling time limitation in here, so at some point even more prey will not increase the amount of food that you can consume. The next important bit here is that we are looking at the stomach. So if you're filling your stomach, there's a certain rate which it takes to evacuate that stomach. And this is often forgotten in models. And that gastric evacuation rate is temperature dependent. So in cold waters, it's actually quite hard to digest something and make room for more food in your stomach. So this is a temperature dependent rate similar to the metabolic rate. So all the energy that is digested, that is the acquired energy, and that in the first priority in the model is used to meet basal metabolic demands and fuel routine behavior. If those demands are met, then energy is allocated to reserves. And reserves in the model are a bit of a buffer because there might be situations where the food intake is lower than the metabolic rate, and then you can use your reserves to buffer that demand. When the reserves are full, then in the model, we allocate all the additional energy to what we call surplus. And surplus in this case could be, for example, energy that can be allocated to growth, reproduction, or migration behavior. When the reserves are running low, this has a direct survival consequence. Fish are starving in that case. But there's another term that affects survival here, and that is, of course, predation directly from the higher trophic levels. So what we are then doing in the model is we are combining survival and surplus to a fitness term here. So in the sense of this model, it has an evolutionary logic already. And the elegant bit is here that we use a dynamic optimization algorithm to run through all these possible combinations. So for every given timestamp in the model, for every given depth step, we ask what is the optimal depth interval to be in for an organism. So we are asking what is the optimal strategy to stay alive and maximize surplus by the end of the season. And then we can go on and can try to confront this model predictions with data. I'm going to show you an example here where we use data from a cruise that starts in the southern part of the Norwegian Sea and crosses the Arctic Circle up into the Greenland Sea up here. Along this transect, we first observe that there is a really steep temperature gradient, so close to the Norwegian coast, temperature around 6 degrees. As we are moving out further offshore, temperatures drop to about 0 degrees up here. We also observe that there's a change in the light environment. So this is surface light intensities on a log scale here. So you can see that the nights are actually getting brighter. As we are moving further north and closer to midsummer, we see that the nights are getting shorter, so we have more light there. And correspondingly, we see that also the acoustic backscatter responds to this. So we see that the nighttime ascent is halted at greater depths as we are moving further to the north. Those of you that are used to looking at echograms might also notice that there are two scattering layers. one shallower scattering layer here, which we know is composed of Mauritius mülleri, and one deeper scattering layer, which is mostly composed of Penthosema glaciale and some of the pelagic shrimps in the system. This is Sagustus and Passifera mostly. So what the authors of this model then, of this data paper then concluded is that based on the mean weighted migration depth here, it seems like these organisms, they are tracking a light comfort zone. So they tend to stay in a narrow range of light intensities that they follow throughout the diel light cycle. And these values are backed up by a global study. So this is data from the Malaspina transect, and we find that the scattering layers also along the Malaspina transect roughly follow this light comfort zone. So predicting the physics in the model, that's the easy part. As you can see, we are able to predict the light cycle quite well, and also the decrease in this, what we call light comfort zone here. So then we can predict the optimal diel vertical migration strategy. So this is the black line that I've overlaid here. And as you can see, the model predicts exactly what we see in the data. We see that the optimal strategy is to stay within this light comfort zone. And the model also allows us to do virtual experiments. So in the reality, we can't turn off this temperature gradient to see what the effect of temperature is. In the model, I can just spread out, homogenize the temperature gradient, and I can see that in the model, diel vertical migration continues. I can still see that the nighttime ascents are halted at greater depth, and I can still see that there's also this deepening in the daytime depth. So if I play around with the different environmental variables in the model, then I can figure out that the nighttime ascents, they are halted because the nights are getting longer, but this deepening in the daytime depth is a gradient in attenuation. So just the waters offshore are clearer than the waters closer to the shore. So this has helped us already to build trust in the model, but also to already understand the system and the cause of diel vertical migration. So we are just at the region where we're moving across the Arctic Circle, but as we are moving further north, seasonality and light regime becomes even more extreme. Woodsole is at about 42 or just below 42 degrees north, I think, so it's not even on that chart here. I'm currently based in Bergen, which is around 60 degrees north, but I've also spent quite some time or lived more than a year up on Svalbard, which is at 78 degrees north. And at those latitudes, the light regime is not just a theoretical construct, it's really reality. You have several months of midnight sun, you have several months of polar night with complete darkness. And this, of course, also affects the animals in this system. So what we were interested in is to run the model across a longer latitudinal gradient. So we are running it from 50 degrees north all the way up to 85 degrees north. And for the sake of this talk, I'm going to ignore the southern hemisphere because, as you can see, the high latitude regions here, there are no real polar marine ecosystems in the south, but the high latitude ecosystems in the north, those are where we have the Arctic Ocean. When we run the model across this very long latitudinal transect, what we observe is that at the lower latitudes, where we have a diel light cycle, the optimal strategy that is predicted is to track that light comfort zone, to migrate into the surface layers at night to feed on copepods, and then migrate down to depths at sunrise again. At higher latitudes, however, during the midnight sun period, it is never really dark near the surface. We don't have twilight in the surface layers. So the light comfort zone is constrained day and night to greater depth. However, in these systems, we have a pulsed environment where we have all the production is compressed to a very narrow season, and we expect most of the copepods that are diapausing during the winter months to be up in surface waters. So the mesopelagic fish are really disconnected from their prey in this environment. And therefore, the prediction is that it is never safe for them to forage near the surface, but in order not to starve down at depth, they will have to take this foraging forays, and they have to leave the light comfort zone and migrate up to surface waters. And this is very risky, because up there, they are vulnerable to predation from larger visual predators. What the model then predicts is that for that reason, the high latitudes are predicted to be population sinks for these diel vertical migrators, while the lower latitudes are population sources in this case. And the barrier here is the midnight sun. So the mesopelagic fish in the model don't struggle during the polar night. This is mostly because of the cold temperatures and therefore the reduced metabolism. So they make it through this dark period, where there's also little food to find because the copepods all, or most of them, are down at depth hibernating. But then what we can do with the model is also to include warming here. So the general expectation is that species are shifting northwards, tracking those temperatures towards higher latitudes. However, what warming would do in the model is it would make the fish more hungry. The metabolic rate would increase, the stomach evacuation goes faster, so they would have to do this risky foraging forays more often, which then also means that the risk of being eaten scales, which in the end would lead to a contraction of the poleward range limit. So I think because the light environment is not affected by climate change, but the temperature environment is, that some of these species at higher latitudes will actually see a contraction, and they are not going to be able to move further north. But of course, we should also challenge these model predictions, and they have been challenged by observations. So we know that there are mesopelagic scattering layers all the way to the ice edge up here north of Svalbard. So these are some acoustic observations during the summer months. And what we can see is there is a dense acoustic scattering layer just below 400 to 500 meters. That's exactly the same depth where we would predict these layers to be based on the light comfort zone in the model. From our ground truthing with nets, we know, however, that there are relatively few of these diel vertical migrators in there. So north of Svalbard, the myctophid biomass is just below 2% of the total biomass. A more recent paper by Shavarsky et al., they show this decline in backscatter, also here in the Davis Strait. So we have the Baffin Bay up north here. And what we can see is that backscatter rapidly decreases across the 66-degree mark here. So they are showing basically the same decline in backscatter that the model would also predict. However, what they also point out in the model, and correctly so, is that this also coincides with a very rapid drop in temperature in this case. So I think that the Davis Strait is a bit of a special case here because it is not only where we see a change in the species composition. So we have a lot of the myctophids south, but very few of them in the north, and there the communities are largely dominated by species that we wouldn't necessarily expect in the mesopelagic zone elsewhere. So lots of fish from the codfish family and snail fishes. The Baffin Bay is a bit special because this is exactly where the polar circle coincides with the polar front. So we have the temperature and the light gradient basically at the same latitude here. But it's maybe not so surprising if we consider the model predictions that the waters that are flowing south from higher latitudes, those are waters that have an advective history from the more northern regions, where we have predicted in the model that because of the seasonal light environment, these water masses would be population sinks. While the water masses, the more Atlantic water masses flowing from the south to the north, they originate in areas where we have a clear day-night cycle, which are predicted sources. Similar case can be made for the North Atlantic in the region where we have run the model. There the case is even more extreme. So we have a branch of warm water that flows up on the west coast of Norway all the way up to Svalbard. But then we have colder waters pushing downwards on the Greenlandic side here. And these water masses actually, they're quite fast flowing, so the water masses flow much more rapidly than we would predict mesopelagic fish to swim. So I think it's reasonable to assume that there is a certain component of advection towards these higher latitudes here. And then, of course, these lines become blurred where the light environment is exported through advection to lower latitudes, or a diel light environment can be exported to higher latitudes with the advection of water mass. Some of the more recent studies have then also looked at the gut content of some of the myctophids up here in the Icelandic Sea, and they have found that some of these had actually quite full stomachs, which might contradict the model at first, but it is not so surprising if you think what they have been feeding on. So they have been feeding on the largest of the copepods in the area, Calanus hyperboreus, and since we predict mesopelagic fishes to be absent from this high latitude, they may be exactly the reason why these large copepods could have been evolving there in the first place. Because only there and then, when the visual predators are absent, then it is a viable strategy to be large and have several years of diapause before you actually become reproductive. So I don't think this contradicts the model actually as much as it seems in the first. So I think the interesting questions that we are not really asking at the moment is where are source and sink areas of mesopelagic fish production and where does this production become available to predators? So we have heard already in some of the last talks that some of these large apex predators, they are able to dive down to the mesopelagic zone and forage directly on the organisms in the twilight zone. But I think there are also areas where this production becomes available through advection. Mesopelagic scattering layers are advected into shallower areas where they then become blocked by shallow bottom topographies. I think this is where we see a lot of the coupling between epipelagic and mesopelagic ecosystems as well. One question that I'm really interested in, thinking about this advection towards higher latitude, to what degree does the advected mesopelagic biomass really fuel this high latitude food system? So far, I have talked a bit about the cause of diavertical migration. So now I'm going to move into the Mediterranean, and we're going to talk for a few more minutes about the consequences of diavertical migration. And I just want to give a shout out to my colleague, Daniel here. He has been really the brain behind this part of the project. We're going to talk about tuna for a second here. We know that mesopelagic fish shy away from the surface waters during moonlit nights. We know that the intensity of a full moon is 2 to 3 orders of magnitude higher than during full moon. And again, we can see from our model prediction really that mesopelagic fish don't enter the surface waters here anymore. And we can just express this as how many mesopelagic fish we would expect to be in the upper 25 meters. And you can see that during the new moon, we would expect lots of them to enter that upper 25 meters, and during full moon, very few of them. And this is also sustained by observations, actually. Then several studies have hypothesized that because of this effect, that there would be a lower mortality in early planktonic stages of pelagic fishes. I think Santiago is in the room today as well, so he has been one of the ones pioneering these ideas already in 2008. But this has been a hypothesis that has been very difficult to test. But I think with tuna in the Mediterranean, we have found a case where we actually can see this effect very clearly. So what we have done is we couple the mesopelagic fish model with a bioenergetic model for the early life stages of tuna larvae, eggs and larvae here. So we predict the egg survival throughout the entire season here. We know already from observation that there's only a very small time window here where actually larvae hatch from these eggs, and this is also reproduced in the model. I have pulled out a zoom down here so for you to see it a bit more clearly, but the onset of this hatching here is really controlled by temperature. Tuna larvae need 19 degrees to hatch. Before that, you won't see any hatching. But then you also see this rapid drop off here after a couple of weeks. And this is also driven by temperature. So the tuna larvae, they have an extremely high metabolism. And in the beginning, these increasing temperatures help to fuel that metabolism and help to fuel rapid growth. But at some point, there comes a stage where we call it a metabolic breakdown. So what happens here is that the tuna larvae have a higher metabolism than their feeding rates, and they can't sustain that higher metabolic rate, and that's when they stop. The little wiggles that you see here in the line, these are just daily variations in the fitness of tuna larvae. For example, because larvae that hatch early in the night, they are exposed to higher predation for mesopelagic fish, while those that hatch late in the night, they have an entire day to grow before they come into that period of first predation. And also because temperature variation and of course feeding rates that are controlled by light environment here. So this is the baseline for our model. But then what we can do is we can add mesopelagic fish predation to this scenario. And what we can see is that both for the eggs and also for the larvae, we can see that there's a huge drop in fitness here, which coincides with the lunar cycle. What we can see here is an order of two orders of magnitude different in larval survival around new moon, when we expect most of the mesopelagic fish to be in surface waters. We can do this for another year just to give you an impression that the lunar cycle is shifting across years, and depending on how the lunar cycle matches up with this very narrow temperature windows where the eggs and larvae can spawn, we would also predict to see some variation in the year class strengths of tuna larvae. This is all good and well, but how well does it predict reality again? I think if we are moving to the field data, so in the Mediterranean, they have been collecting eggs and larvae from surveys for over 2 decades. We have done some statistical modeling on it, and what we can see is that there is actually a 3-day offset. The highest spawning activity is just before full moon, when we expect least of the mesopelagic fish to be actually in surface waters. And the consequences that are predicted by the model was this 2 orders of magnitude difference in predation from mesopelagic fish. So this, of course, begs the question, what other fish could have adjusted their spawning to the lunar cycle? We know that there are a couple of tuna species that seem to have synchrony with the lunar cycle. But I just want to point to eels here, for example. There are several studies that show that eels tend to spawn during new moon, around 150 to 200 meter depth. And again, you could make the contrary argument that we are making for tunas in the surface waters here. Spawning down at this depth means that at new moon, mesopelagic fish really migrate into the surface waters. So they pass through this depth layer very quickly. They spend little time there, which then should be relatively safe for eel larvae, while during full moon, the entire mesopelagic layer would be deeper and it would actually coincide with this depth layer. So I think by using these models and asking some of these evolutionary questions, we can actually learn quite a bit about the system. So I would challenge you all, if you look at diel vertical migration, if you look at scattering layers, to ask also this question, what or how does the presence of mesopelagic fish shape the ecosystem? What could their absence mean for the entire system? And where do we see differences between different ocean basins here?
Hi, thank you very much. That was a really interesting talk. My question is about the dispersal on ocean currents you were talking about. So there's often fairly strong shear in the upper couple hundred meters in these currents. And so when you're talking about these very short distances between 2 water masses, I'm wondering, would there be a substantial shear also in the biological communities that are being transported, with some being transported rapidly and others perhaps that spend more time at depth being transported more slowly?
Yeah, I assume that would be the expectation. I can't say that we have done any of the modeling on it, and we don't have that resolution in the observations either. But yeah, that would be a reasonable expectation, I would say.
I have a question, a comment and a question up here. So your last comment about the eel larvae, I'm not quite sure I believe, because I think maybe from myctophid standpoint, that may be true. The myctophids maybe go up shallower than the 150, 200, but we know acoustically and from trawl and net sampling that the species and the communities stratify vertically, and that we do see quite a few of actually hatchet fish often between 150, 200 meters. So there might be different species that will come up to those depths. And in fact, the real question I had was, it's interesting in the north. Do you see a latitudinal migration, seasonal migration, a horizontal versus? I would wonder why the fish don't take advantage of the summer light and increase primary productivity, maybe potentially secondary productivity in the summertime, and migrate north to take advantage of that.
I mean, obviously, we see this with all the large epipelagic stocks, right? I mean, in those subpolar seas, we have huge herring and capelin populations that do this extensive migrations up north, and they're certainly benefiting from the longer days up there and the production. I'm not aware that we have any evidence for migrations in the mix of it. I mean, maybe Stein knows, but I'm not aware from any of the Norwegian waters that we see this. No.
Hi, Tom. Debbie Steinberg over here. Question for you. Do you think it's possible that sinking detritus helps feed those myctophids during the summer when they're can't be up there in the surface waters? You mentioned that at least one study showed their guts were filled with copepods, but certainly fecal pellets and other things have been found in the guts of myctophids, and it's so productive, and maybe. So has there been much gut content analysis or other things that might show that or test that?
Yeah, I think certainly that could be the case. So in the model, we only have a prey field composed out of zooplankton, but I know depending on how I tweak that field, if I would have a secondary production down deep, that would change the picture slightly. So I think then we could have some survival or higher survival at least during those periods. If it comes to gut content analysis, I think in the myctophids, what we see is mostly that it's dominated by copepods.
Thank you.